Hello. Hello. Um, yeah. Hi all. Welcome to this week's live interaction session. So people are getting joined to the session. I appreciate all who joined on time. Let's wait 2 more minutes so that the others also who got late will join and we can start our session. We have Rahul also here. Rahul has some technical issues, so by the in between the 2 minutes, we'll sort it out. Please wait.
Pallavi, can you confirm I'm audible? Yeah, thank you. Yeah, like Pallavi said, I'll just wait for another 1 minute. We'll start by 5:05. So we have around 20 participants from the Zoom. I think some people have joined through YouTube also. So let's give another 1 minute for others to join, then we can directly go to the today's session. So meanwhile, like we always say, please join through the Mentimeter also. You can directly scan this QR code, or you can directly go to menti.com and give this code so that you can join. Today we may have more than 2, 3 activities over there. Yeah. So after last week's session, I think today you people might have come with some questions and doubts. Okay, let's start the session. So thanks for all who joined. Once again, good evening, everyone. So we have around 21 participants. I'm not seeing much people join to the Mentimeter. Please do join. So it will help us to make this session a little more interactive. Yeah, so let me start with a small introduction. So, a small recap. So, we are in the fifth week of the course, total 8 weeks. In last week's sessions, you might have seen got introduced to the biomolecular proteins, and you might have got introduced to some proteins around us. I think it is activity by Pallavi, where you found some proteins from different samples, I guess. And then you have got introduced to enzymes also. Nice. So this is what we have seen last week. So you anyone have any questions regarding last week's session or the previous 4 weeks or 5 weeks? Any doubts on that?
No,
I
sir.
think okay. Yeah. So I think we got few questions in the discussion forum, but still we are seeing not seeing sorry, that much interaction in the discussion forum. Yeah, we'll come to that later. What about others? Yeah, I heard one knows. Anyone else? Any questions or doubts or any topics you want to discuss among the 23? No one has any doubt? Like I said, it's not necessary to ask questions related to that course. You can ask in general biology also. We can have a small discussion regarding that. If there is no much questions for now, yeah, let me show you one activity in Mentimeter. Like always, it will be a direct question. Start answering the question. We can have a small discussion on that also. So the first question is, so it's a little tricky question. So I just want to you to imagine a thing like you have a sample of poliovirus. The size of the poliovirus is around 30 nanometers in size, and you have a light microscope which can magnify up to 10,000x. The question is, can you see this poliovirus through that microscope? So don't think worry about magnification. So you have 10,000x, sorry, yeah, 10,000x magnification. So don't think otherwise. So just think logically and tell me, like, can you see this virus through that given microscope? If it is yes, just state your answer, like support your answer with a justification of single line. No, also same. At the end, we can discuss the answer. Anyone? I think only 80 of you have joined the Mentimeter, so I would recommend others to join also. Especially those who have joined to YouTube, please join to Mentimeter. Nityasri, you have said yes. Can you answer that in the Mentimeter? Or if you find any difficulty in joining Mentimeter, can you let us know? Not just saying yes, just give me a sentence answer, like how we can see, why you think that we can see, or why you think we can't see. 30 nanometers is too small for magnification. Okay, that is a decent amount of thinking about the logic. Yeah, right. The magnetic is not enough. Okay, others? Any thoughts? Those people who are answering the chat, can you answer in the Mentimeter also? Like, we just want a sentence, not just yes, no. We want a sentence answer. Like, why you are thinking that it can be seen or it can't be seen. Okay, another question answer we got, like, no, it would be blurry as the size of the virus is only 30 nanometer. Okay, you are close. This answer is very close. No, because resolution limit must be at least 200 nanometer according to textbook. Okay, fine. Okay, we can see. Someone is telling we can see. Nice. Oh, it's a very clear answer. Yeah, poliovirus is about 30 nanometer in size. The light microscope has a resolution limit of 200 nanometer because visible light has a relatively long wavelength. Even if you increase magnification to, yeah, I think the word limit stop you. Right. Nice answer. Others, what do you think? Since we have already good amount of explanations, now we can just tell yes or no. Okay, so No, you cannot see. Yeah, the 30 nanometer polyvirus using a microscope. Okay. The person who answered this answer, like a polyvirus is about 30 nanometer in size, and you have mentioned that the resolution limit. That's the right answer actually. So even if you keep on increasing the magnification, sometimes you can't see the things clear, materials or object clearly, because like we already mentioned in our second or third week lectures. So when you want to clearly see an image through microscope, it is not only about size, that is magnification. It's also about resolution, right? Resolution is nothing but distinguishing between two separate objects. So when the resolution is very less, even if you keep on zooming, everything will look like will look blurry, like you people stated, or it will be smudged together. Two objects will be smudged together, and it will look like a single object. You won't get a clear image. So for poliovirus or any object which are in that size range, we need a electron microscope to see them, right? So because the limitation comes from the visible light which we are using in light microscope. So we have to directly go to electrons so that we can get a higher resolution and clear image. Yeah. So thanks for that answer. Specifically this one, it was very nice. Like many of you have thought in that way, in this perspective of resolution. That is good. Fine. So any other have any doubts or comments on this regarding the microscope or something like that? Since you people have already reached a level of proteins, you have already crossed three or four biomolecules, right? No doubts as far now. Any general doubts? Or if you want to know anything more about any topic or something like that, you are being so silent this time also. Fine. So then I think today's session will get over very fast. So the next let's finish the activities today then. Meanwhile, we have a question from this discussion forum also. Pallavi, can you read out that? We can discuss on that. Let her take some time. I think there's some issue with her headphones. Meanwhile, we will finish one more activity in the Mentimeter, since you do not have any doubts. It's very general answer, general question actually. So where do you see proteins in your everyday life? Write one example. Egg, nice. Don't say protein powder. Anything else which will be surprised. Think any kind of proteins. Where do you see proteins in your everyday life? One example. Egg is the most common one. It's nice, nice catch. We have 27 participants here. This can't be this silent. Even Pallavi is silent actually. Any other answers? Those who have joined Mentimeter, please do answer. Spinach, yes. Nails and hairs, yeah, both are proteins. Nice answer. Nail and hairs, nice answer. Milk, yeah, it has decent amount of protein. One more milk. Any other things which is not edible or except food? Anything else? Meat, nice, right? Hemoglobin, yes, hemoglobin is a protein. Pulses, yeah. Nuts and seeds, yes. Other than food materials, anything else where you can see proteins? Where else we use protein? Not just yeah, by having as a food or supplement. Is there any other place we are using proteins? Silk, correct? That's a nice answer. So like that, something anything else? Insulin, cool. Silk is a very good answer. Like that, anything else? Which proteins we won't directly eat, but we come across daily? For example, protease or lysozyme, where it will be present in detergents, sprouts, dal, yeah, all food, vaccines, okay. Collagen, nice. It sometimes we use collagen as supplements, and also it is present in most of the cosmetics also. Keratin is a protein which is present in our hair. Nice. Nice. Hormones and enzymes. Oh, very general actually. So cool. Okay, fine. So you will have some idea. So, but I want you to think in a way that protein is not just food supplement. Whatever the milk, egg, nuts and seeds, spinach. So I am thinking like everyone is thinking protein as a food supplement, or it is only present. We are using that as a nutrients, using that as a nutrient value, but it's not like that. There are other applications for proteins, especially when it comes to biotechnology or pharmaceuticals. There are proteins are being used as many drugs. Uh, Immunoglobulin, we said, right? MABs, those are proteins, right? So there are lots and lots of proteins which we won't eat, but we use that protein for some other purposes. Think in that way. So, like I said, a detergent, some dairy products has proteins. What else? Um, like I said, some cosmetics has proteins. Sunscreens has proteins, right? So yeah, so think in that way. Not only about this protein, when you talk about uh, lipids, everything, we can't just see them as food materials. We are using carbohydrates for other purposes also. Uh, like xanthangum, it is not just a food, like directly food thing. It is an additive which solidifies liquid. That's what we read, right? So it is being used in different applications. Like that, carbohydrate, lipids, proteins, everything is being used in other places also, right? So that's what we want to convey here. So I think now Pallavi will be ready with the question which we got from the discussion forum. Pallavi, can you come on?
Yes, yes. Hi, all. I had some technical glitch was there. So we have a question in the Google form which was shared to you, and Joshita has inquired about. Um, so I will read out the question first. I was just reading about guinea worm disease, and I had a doubt like, since stomach acid digests the crustacean having larvae, how does this larvae also not get digested with it? And how does it survive in harsh conditions? So basically, I appreciate Joshita, even though this is not directly related to our topic, whatever we have discussed till now. But the way you are thinking when you read something, that is actually very nice. So in this guinea worm disease, what happened is like when you drink a polluted water or something, so some parasites will get will reach our stomach, and that is what causes the disease. So this larvae, guinea worm larvae, whatever is responsible for this disease, is inside a crustacean which has a chitin like substance and all. So this outer organism, which is a crustacean, will be digested by the acid which is present in our stomach. So once this crustacean is digested, the larvae is exposed to the stomach environment. But this larvae has a cuticle. Like just now we talked about collagen, right? So this is made up of some collagen and all. This cuticle is not sensitive to the stomach acid. Like that pH is not enough to digest this larvae cuticle. So this larvae will get like won't be digested by the stomach acid, and they survive and they cause the disease. So so if you are thinking in a treatment point of view and all, so you must be thinking like our body itself is trying to fight against this thing, but there are like what demerits all also this points we need to focus more so that we can find a way to destroy this larvae and all. So even though this was not directly to our content, the way you are thinking is like very very nice. I appreciate and I motivate every one of you to think in this way. So even though I was not sure about this, I also just read about it and I'm like conveying it to you. So this way you can read, you can discuss such things here, and we can learn together. Jochita has raised her hands, so we'll hear from her directly. Joshita, can you unmute?
Yes, ma'am. Good evening,
Yeah,
sir.
hi Joshita.
Yeah, so actually, I started reading about parasites recently because I was very interested in it. And a lot of parasites actually survive our stomach acid, right? And they are evolved to survive our stomach acid. I would actually like to discuss about it. Like, how are they doing this? Like, how are they evolving to doing this and all?
Okay, so the one thing is like we know survival of the fittest. So in the process of evolution, whoever survived the conditions has uh, like survived and they are thriving now, right? So one part what I came to know about this particular larvae is their particular collagen. So whatever materials are there to make their cuticle and all is helping them to survive such conditions. So uh, compared when we compare with bacterials also, there is a group of bacteria who can withstand very harsh conditions like their uh, extremophiles are there which can thrive like very saline, very like high temperature, cold temperature, like the different categories are there. So actually like when we see the whole bacteria whole parasite groups, I don't know which are the particular things which helps them to survive. One is their cuticle, which is not sensitive to the stomach acid and all. So it's nice to like, I'll also try to read about it. We can ask Rahul also if he is aware of something else, so that we can fight against this parasite and we can come across different treatment strategies against them. Rahul, do you have any other?
Hi, Joshita. So that's a nice question actually. So,
Yeah.
like Pallavi said, yeah. So not all the parasites can survive this pH, which is the acidic pH in our stomach, like you people discussed. So what happens here is like I want to just Joshita, you're listening, right?
Oh, yes, sir. Yes,
Yeah,
sir.
yeah. So I just want to tell you about, remind you about the sterilization. So what we have done in the sterilization is like we will increase the temperature to 121 degrees Celsius and stay there for 20 minutes and then we'll come down. So we need 20 minutes of time in that temperature to get rid of all the microbes. It is so same way. So if a parasite goes into our stomach and if it needs to be killed, it needs some time of that acidic exposure, right? So mostly in this digestion, it will be like 2 hours or 1.5 hours. So sometimes what happens for some kind of parasites, they have the ability to thrive this for certain amount. For example, just imagine a parasite which can survive for 3 hours, but the digestion process itself, it will go for only 2 hours. So it can reach intestine. Once it's reach intestine, it can go through lots of changes. And the main thing is like how Pallavi said, it can make cyst and it can make eggs. So those can survive this temperature pH, or there only it can sometimes it will get activated only in that pH, in that acidic pH. Until that, it will be very dormant. So to understand that, you may need to understand one thing called spores. In case of fungus and all, so they will make spores, which is like we can't say that those are like eggs. So it is a dormant state. So where all the cellular activity will be arrested, but the cell is still alive. So when they are getting a good environment or a suitable environment for thriving, they will again grow back. So these are the things. So some parasites can go inside our stomach like spores, like eggs, or like cysts, like that. So these are the different ways. Sometimes, like I said, like I mentioned, that is not more. The time is not enough to kill them. Or sometimes what happens, where we are talking about this 80%, 90%, 100% sterility, right? So when a bunch of parasites are going inside, half of them might have got killed, half or 80% or 90%. Even 10% when it reaches intestine, it can start infections. Those also can happen. So these are the different ways I can think of, and not all parasites are going only through mouth. So some parasites can go, you can inhale them. It will go through lungs. Sometimes directly because of the blood contact or something like that, or a cut, it can go through that also. Yeah, so that's what I can think of as for now. You got any idea? Like,
Yes, sir. I got some idea, but I was actually willing that professor would also discuss about this a bit briefly and how we can actually like cure it. So in case you have digested something or something happens to your body and then there's a parasite inside your stomach or inside your body right now, you can't wait for it to be eliminated naturally sometimes because it may be a fatal process as well. So I was actually wondering if we could actually make Professor join and discuss about this if we may, but I don't know if that will be possible right now.
yeah, maybe not today. I may come convey this to sir. If he wants to write something in the discussion forum, let him write. Or if he's very much interested, he may like what I'm telling is like I'll convey this to sir anyway.
All right, sir. But do you guys have any idea about like that type of medicines that can be digested, like taken orally or something like that, injected into your body where you can technically kill the parasite which is inside your body?
Definitely, right. So not only parasite, in a very general perspective. So whenever we are taking some medicine, any antibiotics, it is going inside our body and killing the microbe, right? So same goes for parasites also, right? So why you have that specific question? So there are so many drugs which we can take, and it will kill something in our body. Either it is a bacteria. So when we talk about parasite is not an organism. So we are talking about a organism, and parasite is nothing but a relationship between us and them. We can say the symbiotes, we can parasite. Symbiosis means it will take something from us and will give something to us. So for example, E. coli, we have lots of E. coli bacteria in our gut. So they eat something what we are eating, and they will produce some enzymes which we are using for digestion. So we have a symbiotic relationship with them. When we talk about parasites, some parasites grow inside us, but we don't have any use of it, but they will take nutrients from us. So they are thriving. Maybe some larvae, some cases, some bacteria, some viruses, maybe. So in such cases, we will give some antibiotics or antiviral drugs, everything, right? So parasite is not an organism. So that you should understand that parasite is a relationship between us and that particular organism. If that organism is an bacteria, we can give antibacterial drug. If it is a virus, antiviral drug. And if it's a fungus, it is antifungal drug. So there are drugs which are already available. I
I
want
was
you to
actually
think.
thinking
Yeah.
more in the worms or something like
Okay.
that type of way, and they're actually harmful for us.
Yeah,
They
definitely.
all the nutrients, and then we feel there are some certain side effects for each parasite actually, right? So I was
Yeah.
thinking
So
that type of way.
yeah, so I can see the tapeworms,
Yes,
Rounds.
roundworms.
fluke.
Yeah, yeah. See, all this has inhibitory drugs actually. So only when it becomes uncontrollable growth, then only people are going for surgery and all. So before that, there are drugs which can kill this larvae or its eggs or get rid of the spores, everything. So drugs are yeah. If you search it, it is available. Yeah.
Yeah, so this drugs, I just had a quick search. So these drugs will actually, like, we can imagine also, right? Like if we want to kill something, we need to stop their metabolism or either their reproduction. So there are particular like drugs which can do this. So we will so as we know, stomach acid and all is like first layer defense system with our body. So if those things are not working, then we will have some drug which will particularly target these kind of parasites and slower their metabolism, either stops their reproduction or so basically, so I just saw paralyze the uh, like worm, as you said, worms and all, it will paralyze, so it won't be able to uh, grow further and cause the disease. Like slowly it will die and it will get eliminated from our body. So drugs are already available. So as we know, the drugs may have side effects and all that, we have to think. But the way you are thinking is nice, Joshita, so that we are going deep into whatever mechanism is happening inside and all.
All right, ma'am. Thank you. And specifically, when we were talking about that Guinea worm disease, right? I don't remember actually. I read it that week, but I saw that it could be potentially fatal, and it's not curable in a way. So I would actually like to discuss about it, like how are they specifically making these type of drugs to kill the parasite and paralyze it and all?
Again, so it is not against parasites. So it is about the organism, right? So if you read about in general about antibiotics or about antibiotics or even anti in I would say antibacterial. So what we are doing, the system needs, if it is a bacteria, the system needs some things to thrive. So what we will do, we will give a protein or any enzyme which will hinder that pathway. Same goes for larva also, like larva or worm, whatever it is. It will need some nutrient or something. It will have a fatal pathway. We will try to inhibit that pathway. So that is how we kill any organisms, whether it is whether it's a worm or it is a mosquito or it is a bacteria, whatever it is. So the mode of mechanism, there are lots of modes. So these are few. In case of mosquitoes, just read about how this mosquito repellent which we are using in our home, how it is working. So all these have similar kind of what to say mode of inhibitions. So that's what we use here also. Maybe for since you are very much interested in this particular disease, maybe we can read about that and we can answer in the mail also, Jashita.
All right.
So
All right,
in
sir.
this point, I'll just convey that this is the beauty of like biology. So if you are applying some techniques to kill it or something, first we need to know about that organism. So there we need to understand what all basic reactions or uh, what are the metabolism or things happening inside the organism. Then whatever we can use, whatever techniques we can use to either promote that metabolism or we can destroy that thing. So in that way, the combination of pure biology and application biology will result in like better world, I think. So both are important. So if we want to apply something, first we need to know about the basics of that. So in that way, these drugs and all are developed. So first we need to know about what organism is this, what worm is this, how it is working, how it is reproducing inside the body. So in that way we can target. So whatever it is in biology, it's like that. So I just want to point out this in this moment.
All right, ma'am. I'll look into it actually. Thank you so much. I know it's not related to our course at all in any way, but I just wanted to know. I was very curious to know about it. Thank you so much.
Yeah, nice.
Anyone else have any other questions? Or any other comments on the discussion which happened here? Okay, anyway, thank you, Joshita. So we'll go to the next activity in the Mentimeter. So it is just a fun question. So with that, if people don't have any questions, we can close the session also. So we just want to see how you have understand the bioreactor thing or bioprocessing. So which of your day-to-day activities or instrument or process shows any characteristics of bioprocessing? For example, I have given here a washing machine, which also mixes and agitates. Same thing which is happening in bioreactor. Like that, anything which comes to your mind, which is resembling bioprocessing in any way, kind of sterilizing or maintaining temperature, anything, any instruments or process which is resembling bioprocess in your day-to-day activity, you come across anything. Can you think in that perspective and answer this question? Nitya is saying temperature. Nitya, you want to unmute yourself and elaborate more on that? So what do you mean by temperature? Curd making, fine. Okay. Like that, anything else? Any other instrument which can agitate or mix or maintain temperature, anything? Centrifuge? You use centrifuge in your day-to-day activities? Just simply like things which are rotating, making cheese by churning. Okay, fine. That's close. Anything else? Just think of any other instruments which are we use daily in our home, in our kitchen. Blender, nice. So most easiest answer. Good. Blender is a good answer. Anything else? Any other thing in your home which can maintain temperature? Any other instrument which can separate things, or any other filters people are using at home? Pressure cooker, definitely. Pressure cooker is one of the best answer. It can sterilize things. It can used for cooking. Right? Oven, yeah, we can maintain temperature in that and increase the temperature. Thermoflask, yeah, we can maintain the temperature for a longer period in thermoflask. That is the right one. It's kind of an insulation which we are using around the bioreactor. Those are better in fridge, not get more microbial growth. Yes, because the temperature is not favorable for the normal microbes to grow, correct? Juicer, yeah, it extracts liquid from solid material. That is a very nice answer. So it is we can directly tell this as a downstream processing. We are extracting something from a solid material, correct? Excellent answer. Anything else like what else spins at your home? Any instrument that spins or mixes or grinds? Yeah, the idea was like that only. So we want you to understand the different processing that happening in bioprocess, not only as bioreactor but also other sterilization. Like nice water purifier separates. It's very nice answer. So where we there is a separate filter is present over there for separating many things. For to separate salt, to separate microbes. Nice answer. Water purifier. So it is also a kind of purification we can say, or we can say sterilization. So here the sterilization won't be 100% efficient, but it is a part of removing microbes also. Nice. Think, yeah. So we want you to think in both upstream and the downstream thing, and we want you to be familiar with all the sorts of bioprocessing, like starting from sterilizing, bioreactor setting up, media preparation, all. Like for example, if you are making a curry at home, it is kind of preparing a media, right? So you add water into that water, you add so many other things, and finally make a final solution. So that's what we expected. Power switches, nice thinking. Refrigerator, preservation. Refrigerator is one of the most expected answer. Actually, we can maintain the temperature over there, which is very, very important for bioprocessing. And you have given a example also like preservation. We use refrigerator for preserving. That is also cool. Nice. Yeah, that's the idea. Nice that we got this many responses. So the last question of the day. So all proteins are not enzymes, but most proteins are. So sorry, most enzymes are proteins. So this you might have got in your assignment also. So just tell whether it's true or false here. So you have already seen what are proteins and like what is the function of enzymes. You know now. Just I want to know want to know whether you will know that all proteins are not enzymes, but most enzymes are proteins. What do you think about this? Whether this statement is true or not? So 5 of you have given us true, one is false. If anyone wants you to comment why you are telling true or false, you can use chat box or you can unmute yourself or you can raise your hands. I can unmute you people. 6 trues and 1 false. Okay. Two false. Okay. Others? Nice. So yeah. So in an order of biomolecules, you have seen proteins. Uh, what are proteins? It's a chain of amino acids. And then to tell the answer, enzymes are a subset of proteins, right? So enzyme, the proteins which has a catalytic effect, which act as catalyst in any biological equations or biological reactions are called as enzymes. So yes, all enzymes or most of the enzymes are proteins only. There are non-protein enzymes also, but most of the enzymes are proteins. But all the proteins are not enzymes because there are non-enzymatic proteins also, structural proteins. There are so many other proteins which act as which they have different functions, not like an enzyme. They are not. They don't catalytic any any reactions. For example, if you say keratin, it's a protein which is a structural protein, so it won't act as an enzyme, right? So not all proteins are enzymes, but most of the enzymes are proteins. Nice. So yeah, we are in the edge of our today's session. So anyone has any other doubts which we can discuss here? So this QA from Google Sheet we have already seen. So any live questions? So it is so silent than last week, but still we have 25 participants, but no one is ready to talk or don't have any doubts. It's a little surprising. Fine. So if you people don't have any doubts, then I think today we can close the session. Yeah. Anyway, we are available in Google Sheets and discussion forum where people can reach us. Yeah, Pallavi, if you don't have any other comments, can we close the session today?
Yeah, if no one has anything, then we can close. So I don't know still why you people are so reluctant to ask questions, even though you don't have a valid question or something. You are excited about something, or you got confused with something, like something scientific or something. We can discuss here. Yeah, we have Varsha. Yes, Varsha, please ask your question. Please unmute yourself and ask. Hi, Varsha.
Hi, ma'am. Good evening, ma'am and sir.
Good evening.
That I have written about heart. I have one doubt.
Okay, ask.
Can I say, at a time when the more blood is venous, okay, but not more blood in actual. What of the effects we have, ma'am?
Like you are telling there is less blood in the body, or
Yes, ma'am. Venous have more bloods, but the arteries have less blood. What are the conditions will happen in this kind situation?
okay, so you know what is the like primary function of arterial blood, what it do?
Yes, ma'am. I don't know.
Like, yeah, one main thing is oxygen supply, right? Another thing
ma'am.
is like nutrient supply, and also the blood is the connecting thing going to all places of our body and supplying, and venous will take out whatever waste is there, and again it will eliminate, like, or filter out. So if there is
Yes,
less
ma'am.
amount of arterial blood, obviously the cells won't be getting uh, proper oxygen, proper cell metabolism won't work. So the first thing you will show is like you will be so tired and all. That is, nothing is working out in the body. The cells are like very tired without or with less oxygen and all, and it will develop into different disease conditions. So, like in a very vague way, I can tell this. So there might be some medical terms for this. When you have less blood, either less blood or less hemoglobin and all. So whoever carries this oxygen molecules inside the body is less, then again the same problem will happen. So in very vague way, I can tell this. So in what context you are asking, whether a doctor told or you read something or something?
Yes, ma'am. I read something. They're given that is that condition relative lack will happen. I can't understand why this they given this answer.
Sorry, I couldn't get you.
Question is, ma'am, at any given time there is more blood in the venous system than that of arterial system. The given answer is relative lack of smooth muscles. I not understand about that answer. Can you explain, ma'am?
Relative lack of
Relative lack of smooth muscles, ma'am. Smooth muscles.
smooth muscles.
Yes, ma'am.
Smooth muscles. Uh, I need to check. Either pumping is not happening, like this heart muscles are weak or something. I don't know, like in which context. Rahul, do you have any idea? Like,
No, like what I'm understanding is like Varsha is talking about smooth muscles. Mostly these smooth muscles are heart muscles. So either way, the heart muscle is not pumping very much. So when the heart is not efficiently pumping the blood, wait, I'm echoing. Yeah, when these heart muscles are not properly working, whether it is pumping too much or whether it is pumping very less, these two problems will come. So either the blood will be pumped too much, or there will be less pump, high blood pressure or low blood pressure will come. So that will have lots of issues in our body. So we can't say that the amount of blood increased here. So since the heart is not efficiently pumping, the blood will be accumulated in some places, but it won't reach the other places. So that's what you are meaning. That's what she's meaning, I feel. So the volume of blood is not increasing here. Just the adequate pumping is not happening. So in such cases, the low pressure will come. All the muscles won't get, all the muscles or all the organs won't get proper oxygen and nutrients. It will directly affect the brain because the brain is very sensitive for low pressure. So starting from confusion, fainting, it can be lethal also. So that's how I'm understanding Vasa's question.
Sir,
to
I
add
had
to
one
it,
doubt
please, Varsha.
that I have an idea, ma'am. Whether the blood will goes backward in venous blood in venous.
No, like we have a very good system which will prevent this backflow and all. No, like we have four chambers for heart and all. There are valves. So altogether, if everything is properly functioning, won't be a black backflow. But if things fails in our body, condition changes. Normally that is not possible. So like adding to your point about smooth muscle word, as you have mentioned, it can be either, blood vessels will dilate and constrict, right? So this also will help in moving blood throughout your body. So if there is no much smooth muscles, or there is if your smooth muscles are not functioning properly, that movement also will be what I can say, limited. So which will also affect the proper circulatory system. So basically the thing will be oxygenate blood won't be getting supplied, and the deoxygenate, like in very vague way, the blood containing waste materials will be accumulating in your body.
Now I understand that question, ma'am. Thank you, ma'am, for your explaining.
Thank you, Varsha. Yeah, so I'm not seeing any other raised hands here. I hope others are clear with the concept. So yeah, just want to remind you once again, start using this discussion forum because that might help you in your final exam also. Yeah, anyway, if you people don't have any other questions, we can close the session today. We can meet you to, next week on our sixth meet. We still have 3 more weeks to meet. Let's see how it goes. Anyway, thanks for who and all joined. Thanks for Pallavi. Pallavi, if you have a say.
Yeah, just like some of you are interested to meet sir and have a live interaction. So if we feel that like everybody is like very actively asking and all, we can we will convey to sir that students are eagerly waiting for you and all. So we encourage you to participate in live interaction. I think none of you are like very afraid of us and all. We believe that we are not making you tensed and all. So feel free. this is our platform to interact, clarify our doubts, or increase our confusions and all by discussion and all. So then only we will go and read, right? So feel free on that. Joshita?
Yes, ma'am. And sir, if we could have a discussion about that parasite thing, I would be very eager for Professor to join actually. Could we make that possible?
Yeah, Joshita. So what I would recommend is like, why don't you directly write a mail to professor? So through the discussion forum, just state directly to professor and write your query. So let him take a call on that.
All right, then. How can I actually approach that? I only did it through the discussion forum last time. I don't know how I can
Yeah,
directly.
in the discussion forum itself, write directly to the professor. Just
All
state,
right.
yeah.
Yes, and again, I'm so sorry. I read about the mosquito repellent thingy.
Mm-hmm.
And it's like a type of chemical is being vaporized from the vapor type specifically. I read about it. And then there's a chemical inside that is present. And then this type of cotton wick thing is going to release it into vapors and it paralyzes the mosquito's neural system from the Goodnight brand. And I don't know why specifically you told me to read out about this. I'm so sorry. I did not catch you at that moment.
Okay, so what happens here is like when we are talking about this antibacterial drug, antiviral drug, we are imagining that some drug which is going to the bacteria and destroying it. But that is not happening there. It is simply, it will inhibit something which is very like required for the bacteria to survive. For example, in case of mosquito, it won't directly go and destroy the mosquito, right? It will directly go and inhibits its neural pathway, so it can't survive. Same way only this antibacterial drug, antifungal drug, or any antiparasitic drug will work. So that's why I wanted you to understand, to just to give you an introduction about what is this mode of inhibitions. So each drug has this one, whether it's a cancer drug, whether it's an antibacterial drug, antiviral drug, everything has a mode of inhibition. So if you read about that, you can reach a point to understand the way you were asking specifically about that parasite. So like if you find a drug for any other parasite, just start reading about its mode of action. If you know the mode of action, then you will come to know why can't I use this mode of action to the particular parasite which you were talking, right?
All right, all right.
Yeah.
Yes, I'm picking
So
up.
if there is a drug which affects one pathway in bacteria, if that same pathway is available in some other fungi also, we can use this drug there also. But if that pathway is not available, then we can't use. So same thing goes for all the organisms. So if you start reading about other parasites, where how they are killing it, and then you compare that with the your parasite of interest, that will help you to do that.
All right, then. Thank you so much.
Sure. Thank you. Yeah. So yeah, let's close the session for today. Let us meet you for the next week. Thanks for who joined.